Exposure to air pollution before and after birth may affect fundamental cognitive abilities. A growing body of research suggests that exposure to air pollution in the earliest stages of life is associated with negative effects on cognitive abilities. A new study led by the Barcelona Institute for Global Health, ISGlobal, a center supported by La Caixa, has provided new data. Exposure to particulate matter with a diameter of less than 2.5 micrometers (PM2.5) during pregnancy and the first years of life is associated with a reduction in fundamental cognitive abilities, such as working memory and executive attention. The study, carried out as part of the BREATHE project, has been published in Environmental Health Perspectives. The objective was to build on the knowledge generated by earlier studies carried out by the same team, which found lower levels of cognitive development in children attending schools with higher levels of traffic-related air pollution. The study included 2,221 children between 7 and 10 years of age attending schools in the city of Barcelona. The children's cognitive abilities were assessed using various computerized tests. Exposure to air pollution at home during pregnancy and throughout childhood was estimated with a mathematical model using real measurements. The study found that greater PM2.5 exposure from pregnancy until age 7 years was associated with lower working memory scores on tests administered between the ages of 7 and 10 years. The results suggest that exposure to fine particulate matter throughout the study period had a cumulative effect, although the associations were stronger when the most recent years of exposure were taken into account. Working memory is a cognitive system responsible for temporarily holding information for subsequent manipulation. It plays a fundamental role in learning, reasoning, problem-solving and language comprehension. Sex-stratified analysis showed that the relationship between PM2.5 exposure and diminished working memory was found only in boys. As yet, we don't understand what causes these differences, but there are various hormonal and genetic mechanisms that could lead to girls having a better response to inflammatory processes triggered by fine particulate matter and being less susceptible to the toxicity of these particles, commented Ioar Rivas, ISGlobal researcher and lead author of the study. The study also found that higher exposure to particulate matter was associated with a reduction in executive attention in both boys and girls. Executive attention is one of the 3 networks that make up a person's attention capacity. It is involved in high-level forms of attention, such as the detection and resolution of conflicts between options and responses, error detection, response inhibition, and the regulation of thoughts and feelings. Whereas previous studies in the BREATHE project analyzed exposure to air pollution at schools over the course of a year, this study assessed exposures at the participants' homes over a much longer time, from the prenatal period to 7 years of age. This study reinforces our previous findings and confirms that exposure to air pollution at the beginning of life and throughout childhood is a threat to neurodevelopment and an obstacle that prevents children from reaching their full potential, commented Jordi Sunyer, Childhood and Environment Program Coordinator at ISGlobal and last author of the study. More than a protein factory, a role for ribosomes in regulating human gene expression. Researchers from the Stowers Institute for Medical Research have discovered a new function of ribosomes in human cells that may show the protein-making particles' role in destroying healthy mRNAs, the messages that decode DNA into protein. For a long time, many people have viewed ribosomes as a passive player in the cell, a molecular machine that's just producing proteins, says Stowers assistant investigator Ariel Bazzini, Ph.D. Now there's growing evidence that ribosomes regulate gene expression, including in human cells. These findings, which were recently published online in eLife, could lead to further understanding mRNA's role and the causes of gene misregulation in human diseases. In the process of translation, ribosomes make proteins by serving as the site for the biological synthesis of them. Specifically, a ribosome reads codons, sets of 3 consecutive nucleotides, in an mRNA message to determine which amino acids to add to the growing protein chain. As part of this process, ribosomes also act as quality control, triggering the destruction of improperly made mRNA. A growing body of evidence has shown ribosomes also play a role in affecting the stability, life, of properly processed mRNAs, thereby acting as a key factor in modulating mRNA stability, level of mRNA, and protein production. This had been shown in organisms such as yeast, E. coli, and zebrafish. In this study, researchers showed that ribosomes affect mRNA stability in human cell lines as well. We're seeing that the amount of gene expression is a combination of mRNA production, transcription, and stability, Bazzini says. Think of a glass of water. To get an idea of how much water the glass holds at any particular time, it's important to know how much water you initially pour into the glass, but also important to know how much water you drink. The same is true with mRNAs. You can measure how many mRNAs are properly made, but if you don't know how many of them are being broken down, how do you really know how many there are? These findings open the door to two exciting research avenues, says Bazzini. The first is better understanding how ribosomes trigger mRNA destruction, the molecular mechanism of which is still unknown. Ribosomes may not be the passive player researchers have long thought them to be. Similar to ribosomes, molecules called tRNAs, or transfer RNAs, are also fundamentally involved in protein synthesis, says first author on the report and Stower's predoctoral researcher Chushuang Wu. We think that tRNAs, which recognize codons in mRNA and provide corresponding amino acids to ribosomes, might have a strong regulatory role in development and in human diseases. The second research avenue is looking at how this newly discovered regulatory molecular mechanism may be related to genes associated with human diseases. Sequencing of human genomes has shown that individuals sometimes have a silent mutation, which is a change in DNA sequence and codon that does not change the amino acid makeup of the resulting protein, as many amino acids are coded by multiple codons. However, the silent mutation might still have an effect if it leads to ribosomes destroying healthy mRNA. One of the most basic concepts of biology is how genes are regulated and how those regulations drive cells to become specialized. We're interested in studying how post-transcription mechanisms work and, in particular, how ribosomes trigger mRNA destruction, how they trigger or recruit factors to carry out this process, Bazzini says. Understanding how translation affects mRNA expression at a molecular level allows us to start thinking about how mRNA translation might shape gene expression in cancer, aging, or viral infection. The work was funded by the Stowers Institute for Medical Research. Mortality risks among pro athletes. Professional football players appear to have a somewhat elevated risk of death, including higher risk of succumbing to cardiac and neurodegenerative diseases, compared with professional baseball players of similar age, according to new research. The findings raise important questions about sport-specific differences in disease development and the mechanisms that drive the elevated risk. Such mechanisms mandate further research, the investigators said. This particular study was not designed to elucidate the reasons for the elevated risk seen in NFL players, nor was it intended to examine the mechanisms that fuel disease development in these athletes, the investigators caution. The analysis, the first head-to-head comparison between elite professional athletes of similar ages, is published May 24 in JAMA Network Open. The work was conducted by investigators at the Harvard T.H. Chan School of Public Health, Harvard Medical School and Spalding Rehabilitation Hospital. The findings are based on a retrospective analysis of death rates and causes of death in 3,419 NFL National Football League and 2,708 MLB Major League Baseball players over more than 30 years. There were 517 deaths among NFL players and 431 deaths among MLB players between 1979 and 2013. The difference translates into a 26% higher mortality among football players compared with baseball players. NFL players had a nearly threefold greater likelihood of dying of neurodegenerative conditions, compared with MLB players. They also had a nearly 2.5-fold risk of dying from a cardiac cause, the study showed. There were 498 deaths stemming from cardiovascular causes among the NFL players and 225 such deaths among MLB players. The study identified 39 deaths from neurodegenerative conditions such as Alzheimer's disease, ALS and Parkinson's in NFL players and 16 such deaths in MLB players. There were 11 suicides among the NFL group and 5 in the MLB group. Extrapolating these differences into absolute numbers, the researchers said, the elevated risk would translate into 1 additional death from a neurodegenerative disease per 1,000 NFL players by age 55, rising to 11 additional neurodegenerative deaths by age 75, compared with MLB players. Cardiovascular causes would account for 16 additional deaths per 1,000 NFL players by age 55 and rising to 77 additional deaths by age 75 in NFL players, compared with MLB players. Even a single preventable or premature death is one too many, so it is critical for scientists and clinicians to pursue further research into teasing out the reasons behind this increased mortality, said study senior author Mark Weisskopf, the Cecil K. and Philip Drinker Professor of Environmental Epidemiology and Physiology at the Harvard Chan School. However, it is important to remember that the actual number of additional deaths from neurodegenerative diseases among football players remains low. The number of excess deaths, 77, stemming from cardiovascular illness was markedly higher, which may be due to several factors, including higher body mass index among football players and the sheer prevalence of cardiovascular illness, the researchers said. Indeed, cardiovascular disease is the leading cause of death in the United States and worldwide. The results point to the existence of sport-specific differences in sport-related injuries and athlete conditioning as important contributors to disease development. For example, head trauma, repetitive head injuries and subconcussive blows in NFL athletes may contribute to the onset of pathologies that eventually lead to certain neurologic conditions, the researchers said. Football players' larger overall size and greater body mass index, which renders them more susceptible to hypertension and sleep apnea, can also be contributors, the researchers added. For their analysis, the researchers used vital statistics obtained from 2 national databases, the National Institute for Occupational Safety and Health for information on NFL players and the Lawman Baseball Database of MLB players, an index combining data on MLB and predecessor leagues no longer in existence. The researchers then cross-referenced death outcomes from these 2 databases with information obtained from the National Death Index, a federal database that tracks causes of death occurring within the United States. Past research has compared health status and mortality outcomes between professional football players and the general population, but such comparisons are inherently flawed because football players are overall healthier than members of the general population. Past comparisons between NFL players and the general population have showed a lower overall mortality rate among football players but an increased risk for death from neurologic disease. In a somewhat more meaningful comparison, a recent study analyzed death outcomes among NFL players and replacement football players who played during an NFL players' strike. This research found a small but statistically insignificant increase in death rates among NFL players. A comparison between 2 groups of professional athletes with similar physical characteristics, levels of conditioning and overall health status can tease out more meaningful differences in risk directly attributable to playing one sport versus the other, the investigators said. Understanding whether and how certain sport-specific activities may fuel the development of diseases is essential to informing appropriate interventions, the researchers said. A life in pro football may have lifelong consequences, particularly in the domains of cardiac and neurologic health, said Ross Zafante, the Earl P. and Ida S. Charlton professor and head of the Department of Physical Medicine and Rehabilitation at Harvard Medical School. Elucidating sports-specific aspects of disease development is critical for our ability to inform best strategies to maintain optimal player health and prevent disease, Zafante added. This study illuminates the importance of former players taking an active step in seeking a comprehensive health evaluation from their doctor. They should ensure they are closely monitored for both cardiovascular and neurological issues, some of which may be treatable. Drug-resistant infections, if you can't beat them, starve them, scientists find. How do you fight a fungal infection that is becoming increasingly resistant to medicine? By starving it, found a team of University at Buffalo and Temple University researchers. To treat Candida albicans, a common yeast that can cause illness in those with weakened immune systems, researchers limited the fungus access to iron, an element crucial to the organism's survival. They did so by using defersirox, a medication used to treat blood disorders. Tested in mice, the results were promising. Investigators decreased iron levels in saliva by 4 times, which altered the expression of more than 100 genes by the fungus, diminished its ability to infect oral mucosal tissue and caused a 2-fold reduction in the organism's survival rate. In the absence of novel drug candidates, drug repurposing aimed at using existing drugs to treat diseases is a promising strategy, says Mira Edgerton, DDS, PhD, co-lead investigator of the study and research professor in the Department of Oral Biology at the UB School of Dental Medicine. Edgerton, along with Sumant Puri, Ph.D., co-led investigator and assistant professor in the Kornberg School of Dentistry at Temple University, published the study in March in Antimicrobial Agents and Chemotherapy. Currently, only 3 major classes of clinical antifungal drugs exist. However, fungal drug resistance has steadily increased and no new classes of antifungals have emerged in decades, says Edgerton. Candida albicans, a fungus among the group building resistance, is the agent behind a number of infections. They include oral thrush, a yeast infection in the mouth identified by a white film that coats the tongue and throat, causing painful swallowing, and denture-related stomatitis, a fungal infection that affects nearly 2/3 of U.S. denture wearers that causes inflammation, redness and swelling in the mouth. The yeast is also the 4th leading cause of hospital-acquired bloodstream infections, which often have high mortality rates, says Edgerton. Candida albicans is the most abundant fungus in the oral microbiome and relies heavily on saliva as a source for essential elements. Iron, the second most abundant metal in saliva, is a critical nutrient used by the fungus in several cellular processes, including energy production and DNA repair. In mice, the group added defuroserox to drinking water to lower iron levels in saliva and reduce the availability of iron needed to sustain an infection. The investigators found that Candida albicans in the mice who received the treatment were less likely to survive attacks by the immune system, subsisting at a 12% survival rate compared to a 25% survival rate in mice who did not receive the treatment. The therapy also altered the expression of 106 genes by the fungus, a quarter of which were involved in the regulation of iron metabolism, directly regulated by iron or had iron-related functions. The study is the first report of iron starvation affecting gene expression of Candida albicans in real time during live infection, says Puri. Other research has shown that treatment with defuroserox does not result in iron deficiency in adults with normal iron levels, forming the potential for preventative treatment for those who are also vulnerable to mucosal infections, says Puri.